Ladies and gentlemen, dear colleagues, it's my pleasure to be today with you and to share with you monoclonal antibodies targeting epithelial-driven proteins like IL-17C, IL-33, and TSLP in patients with atopic dermatitis. These are my conflicts of interest statement, research support, consultancy, lectures, and also member of scientific advisory boards. Let me start with that where we have a lot of us interest on atopic dermatitis and the role of IL-33, 17E, and TSLP released from keratinocytes directly or indirectly. How do they support the type 2 inflammation in patients with atopic dermatitis? And our knowledge is increasing every day. And I will start with the first candidate, IL-17C. You're probably aware of the compound called MOR106. This is fully human recombinant IgG1 monoclonal antibody. This is the first in class for gas IL-17C, which is it was in a clinical development for atopic dermatitis. And the mode of action of IL-17C, it's a specific potent inhibition of IL-17C. And it is important to know that IL-17, it is particularly important in a different cytokine pathway, which synergistically activates the pathway, especially in type 2 inflammation. The rationale was there for atopic dermatitis, of course, because all these cytokine alarmins are playing a very important role in atopic dermatitis. And once the first trial was done, it was very encouraging, looking at a mean percentage change from a baseline. So it was roughly 50-60% with more. It was a little bit also with placebo, and it was interesting that after 4 weeks of the treatment, it was still improvement of the signs and symptoms of atopic dermatitis. And also, if you look on the EASI-50, it was dose dependent, that it was shown that the highest dosage is having the best response. So far, so good. Everything was fine regarding the safety. Anti-IL-17C expressed that what we expect. It was well tolerated. So then the question was, okay, we should go in the phase 2. And then something happens. You know, IL-17C studies, clinical development was terminated because for futility. In the interim analysis, the data didn't show that what was expected. It was too pity, but you know, we have to go on. And we are moving to the next target. And the next target, as you can see, suddenly everything changed. It's not anymore IL-17C, it's IL-17E, also called IL-25. And then you see IL-23, IL-33, and TSLP. And IL-33, it is a member of IL-1 family of cytokine. It's expressed at the high level of the epithelial cells, and especially in patients who have a filaggrin deficiency. You have different noxes like pollen, house dust mite, animal horse. You may have helminths, scabies, viruses, bacteria, fungi. A lot of the different noxious can activate the production of IL-33, and IL-33 is having a direct effect on inflammation. It's known with an important role in induction of type 2 immunity and the resolution of inflammation. So it was thought this is probably an ideal target. And as an ideal target, the phase 1 studies were done, and they have been also presented and also published. What you see, it's an average decrease of the EASI of roughly 50%, which is not bad. You see the number of the patients who are achieving an EASI 50 and EASI 75. It was not bad. It was very good. If you see 80% of the patient are achieving an EASI 50, this is something that is probably also known from the previous trials. Number of the patient was very low. However, this was very promising. Promising also, if you see the absolute EASI is also decreasing. Why? The patient have been treated with IL-33 inhibitor. You probably know the name. It's called etokimab. And for all the Europeans who love SCORAD, the absolute SCORAD also decreased. And this was in the phase 1, the proof of concept trial. It was even statistically significant, highly statistically significant. Moving further and looking to the investigator global assessment, okay, it was good from a severe to, let's say, a severe one. DLQI, 50% improvement. Age was good, but not so good improvement. However, promising. What happened is another company has also done another anti-IL-33 receptor. It's called CNTO7160. And also another company, Regeneron, has also the same target. And what do we see with the highest dosage? It is roughly in a decrease of 20 to 25% change from baseline in score. So IL-33 we thought at least it will be in a good target, and as a good target, it was interesting to observe what will happen. And the phase 2 trials failed the primary endpoint. Very disappointing to learn that IL-33 failed to meet the primary endpoint in a phase 2b. Therefore, the studies have been stopped in atopic dermatitis. It was too pity, but this is the way how we also learn. We learn IL-33 alone is probably not the only target. And finally, probably anti-TSLP antibodies will be effective. Similar like IL-33 and also IL-17E. You see the same trigger. Even Staphylococcus aureus is having more potent effect. TSLP is a member of IL-2 family, and it is originally identified as a molecule that can stimulate murine thymocytes and promote B cell proliferation and development. T cell are also involved, but these cytokines are driven from mainly epithelial cells, like for example from the skin. And you see different mode of action. One of that targeting type 2 immunology, type 2 disease, like Th2 cells, where we have a direct effect probably on the production of IL-4/13, but also IL-5, which might be important in asthma, or also in innate lymphoid cells of type 2, where we have also probably in a direct effect, and also indirect way through the Langerhans cells. So we are learning it could be in a target, and the cytokine which is driven from epithelia. What is happening? You see the compound, tezepelumab, and also another one. They are on a development for the moderate to severe atopic dermatitis and uncontrolled asthma. The recent publication was shown that tezepelumab could be in a treatment option. You see the patient who were treated with tezepelumab plus topical corticosteroid and compared to placebo plus topical corticosteroid. EASI 50 responder rate was good, but the problem is always that also placebo topical corticosteroid are achieving a very high response. Therefore, it was not a very significant, especially if you look, for example, in the patient, the percentage improvement from baseline compared to the patient who are receiving placebo and patient with tezepelumab. Interesting was a nice probably response on pruritus, where it was significant, it was achieved, but not at every time point. We see some separation between the placebo and tezepelumab, but not really something which is probably also clinically relevant. Regarding safety, there were no safety issues among the placebo plus tezepelumab or without tezepelumab and topical corticosteroids, whatever group we were looking for, very similar safety profile. So it didn't have any big problems in the safety, but the problem was that the primary endpoint were also not reached in atopic dermatitis, and this is why the company suspended a phase 2 trial for atopic dermatitis. Very pity. Very pity for 17C, pity for IL-33, pity also for TSLP. But, you know, sometime we are learning. We are learning that probably the target is not atopic dermatitis. It could be that the target is asthma. And it's not mandatory that if something is not working in atopic dermatitis, is not acting or not working in asthma. So probably TSLP will go in the other direction, development for the patient who are having asthma. You see results in the patient who are having a low eosinophil count, a high eosinophil count, Th2 status, high and low, whatever. In all these groups, tezepelumab was shown to be effective in controlling asthma. So in asthma, it will probably make a breakthrough and it will be a treatment option for this patient. With this, I think it's very difficult to make a conclusion. So next lecture that I will have probably will be revisiting therapies for atopic dermatitis that failed clinical trials, in fact, than cytokine-driven studies from epithelia. Or you can do it in a different way. The show must go on. This is what my colleague from Hanover, Thomas Werfel, is mentioning, so you can continue. And I think we'll not stop with this one. So there are new treatment options that will come in the future, new target. And I'm very pleased that I was able to show you at least one part of the clinical trials that were done in the past regarding these specific cytokines. Thank you very much for your attention.